Richard. I know it's always awkward when the company writes you a bio and to read it out. And when I listen to it, I said, it sounds so alien. It's true, but it's so fabricated in a way. So I'm leading the secondary pharmacology group in Novartis. And we start early safety assessment and then continue into the clinic. So what I'd like to talk to you about, and this is really a last-minute drop-in for a full presentation, it's not just the iPSC-derived cardiomyocytes, but in general terms, what SIPA is concerned about and what we are in the industry particularly concerned about, how we can make a better pro-arrhythmia assessment. And in that sense, I think if you look at the landscape, to start with, everybody is clear that we need integration of ion channel effects, and it's absolutely necessary. We have clinical evidence for that. Luckily, some of the compounds which were before HERG assessment introduced to the clinic went through, and they have obviously various ion channel effects, and they survived, and they are still in the clinic. And we have evidence for those compounds like, for example, the Novartis compound Tasigna, when there is a very strong signal, HERG signal, but because the therapeutic index is reasonable, it goes with a box warning, but very rarely, or in terms of torsades, never happened during the clinical phase because the monitoring is in place. But what we recognize that we need cardiomyocyte-based assays, and I think there are three criteria we agree upon: human, ventricular specific, and mature. With the middle one can argue how the combination should be. And then the potency of the other major element, I think, in terms of pro-arrhythmia assessment in the preclinical setting is the potency of ion channel inhibitors is not well correlated to clinical arrhythmogenicity, again, banality at the moment, but need to assess channel kinetics. I'm absolutely convinced that if we go after potency again and again and again, and it doesn't matter now we have 150 compounds properly studied, and we add another 50, it would give you a tremendous input. I think what would help if we go by novel experimental approaches, and the in silico models should be aligned or married with the in vitro models. And here we don't discuss any of the possible animal experiments preclinically. Certainly I will focus on the other elements, the in vitro. And the definition of preclinical biomarkers for proarrhythmia is something which doesn't have to be a single readout of the potency or a couple of ion channel potencies, but could be a fingerprint which doesn't exist at the moment. So the background, the assays which we are using, which are using stem cells or iPSC-derived cardiomyocytes represent immature cell population, full stop. And we can argue about it because many of the CROs would maintain that these cells are absolutely perfect. At the moment, yes, they are very close to that. But a very simple thing is, which I will represent here or present here, that once you get it into practice, there is by far no perfect iPSC-derived cardiomyocyte which is available or would be available for routine experiments. There are very nice studies out there, and there is a huge value to that. But using it for regulatory, it's by far not ready. So decreased electrophysiological stability is, if you go through the presentations with a critical eye, it pops up all the way. Hence, drug effects are biased. Ion channel components of early after depolarization formation could be distorted. Functional and structural features do not resemble mature adult cardiomyocytes. And that includes also that the maintenance of these cells, and we need, for example, exposure for weeks and months, for example, with cardiomyocyte studies done for protein kinase inhibitors, it's a must that we have stable cells. And the stability of adult characteristics is poorly sustained, as I mentioned. The present in vitro cardiosafety assays have certainly limited predictive value. It's nobody to blame about it. I think what Will Redfern produced and the people who were associated with the paper in 2002 did a phenomenal job. If you look at what is going into the clinic, it's almost extra safe. You cannot say it about DILI, for example, or some of the other toxicology effects. But we also recognize it's overtly safe. So probably we lose lots of compounds which should go into the clinic. And pro-arrhythmia assessment basically should focus on changes in ion channel kinetics. No integrated approach exists for multiple drug effects on cardiomyocytes. And there is a need for mature cardiomyocyte system capable of generating reliable and consistent results. So you would say that this, I was reading out SIPA so far. But the one interesting thing with SIPA, that SIPA produced enormous amount of data, made great progress, but it's not there to do regulatory. And we had a discussion with FDA, which was late last year, and I raised the question that we were at the meeting when FDA first basically raised this issue about SIPA, and they were pushing very hard stem cells. And at that time, there was absolutely no reason to do that. So what people considered that, okay, this is already ready-made, so we have the cardiomyocytes from iPS cells, so let's do the experiments. And I think lots of energy went into that without making sure that these cells are stable and similar to adult cells. This is a little bit of repetitive slide. I really just wanted to show the simplicity of how we can approach it. So go to mature iPS cardiomyocytes, and then these are the different elements which come up here. The signal detection, the modeling and simulation, structural cardiotoxicity, contractility, and disruptive assay technologies would all be in the same bucket if we are aiming for a comprehensive assay. And this is materializing during SIPA, but we also initiated an IMI project which asked for substantial support. And it's very difficult to achieve it, because certainly this one would wholly focus on the maturation of the particular cells. Therefore, it's not just cardiomyocytes, but what is in the iPS cell field new, and how would it be implicated or applied to the cardiomyocytes. So the outline is very simple, and I think everybody, when I talk to people, and this was given to me by someone who is developing in Novartis the iPSC-derived cardiomyocyte and other cells. So they have a pretty clear protocol, included the QC processes, the iPS cell line development, the pluripotency verification, differentiation, potential characterization is in place. And then basically the cardiomyocytes had to be characterized, which is regularly done, but that's where we find out that not all the features are perfect. And that shows up in different assays. When we look at the considerations, the reproducibility, the characterization platforms, which should combine genetic and functional characterization to fully define maturity. The very first sign when these cells became available was that these cells are beating spontaneously. There could be two reasons. One, that they have pacemaker cells included. Second, and that's why they have spontaneous rhythmic activity. The second one, that these cells are on their own active. Now, if you are working with cardiomyocytes, adult cells, they are not spontaneously active. They need a stimulus. And this gives you an idea, and we all know that who do electrophysiology, that the baseline, which in this case the resting membrane potential, is not absolutely stable. And one reason why hERG is so difficult to study in these cells, because hERG contributes to the maintenance of the resting potential because of the missing or somewhat missing element of Kv2.1. So what people try to achieve is, just very briefly, I put it together here. Again, this slide is coming from the cell culture group, that they basically put together all these channels which would be important to study in terms of the action potential generation. And then they characterize the expression of these cells in ventricular, left and right ventricular signature, and also in the whole heart. So the whole heart was the baseline. And then if you look at the expression of various channels, that you see differences in between left and right ventricles. And this is basically an in-house cell line which they developed. And you can see that this is 14, 21, and 28 days. And they try to analyze basically what would be the best time to study these cells for arrhythmogenicity. But you see immediately that this is changing in their hands. So if we go ahead with time, there will be different expression without doing any particular thing to these cell cultures. So what an electrophysiologist, I would like to see that this is remaining steady. And if somebody is overexpressed, but it is acceptable or underexpressed, and it is acceptable, remains the same during a brief period, at least a brief period of time. And I think that's where the problem starts, when you get a cell line and then it is given that you do the experiments between day 14 and day 16. In your hands, it might be day 21 and then day 25, which gives you the best results. It really depends on the labs. And that makes a difference in between how do we compare these data. So is it one company which should do all the experiments for all the industry? That certainly is not a solution. So what we were doing, and I wanted to highlight this paper by Bob Perlstein. It was published very recently. He was looking at the different characteristics of the action potential and tried to pull now, because we are dealing with the cardiomyocytes, all the necessary components which could basically contribute to the triangulation phase, where we know that early after depolarizations could happen. And the one reason was that, two reasons basically. The one that this segment is here between -30 and -50 or so is really crucial, where the action potential can spend more time or less time in this particular segment. That could make a difference. And also it could make a difference that a particular channel blocker, what kind of kinetics we can assign to a particular channel blocker. And they came up with this idea. This is nothing new. Many years ago, the Viennese group, Professor Höring's group, published that there are two types of hERG blockers in his belief. One is trappable, and the other one is non-trappable. So the trappable, basically designed by the kinetics of the ion channel, could get trapped in and accumulates. So very fast could basically lead to a proarrhythmic effect. And the other type would dissociate and associate so dynamically with the channel. Now, this was done in oocytes. And it was repeated in our lab as well for CHO cells when the hERG channel was expressed. So one idea was that we could repeat it in cardiomyocytes and see what happens. But bear in mind that many of the compounds which have a strong channel effect at hERG have also other ion channels. So the combination of these effects is, with the combination of immature cells, is really something which makes it extremely difficult to study at the moment. So this is basically a model cell which shows how a non-trappable compound does not cause proarrhythmic effect, while another one would basically produce the early after depolarizations. The model is something which everybody is using. This is the O'Hara-Rudy. But the one big difference between the O'Hara-Rudy or the Markov, this is the combination of the two. And then I show you why. So this is basically an alternative model. So what we try to achieve with this model is somewhat different from previous efforts. We are very much aware, and we know the FDA efforts and other efforts in academia alongside this. So what you see here is basically a recording of action potentials from cardiomyocytes, EPS-CCMs. And just ignore propafenone and see the basic action potentials here. And they were just marked with the different currents. But here, this side is the interesting one. So this is the action potential recording without any hERG blocker. And this is how far we got before we had to break this project. What we try to do, basically, once we digitize the action potential, to feed it into the model. So you go to Notochord, you get the digitized data, and then you enter all of these data, so basically the action potential, into the model cell. And the model cell does something which is quite nice. You can see these different lines. So this is still the action potential, and in between, there are three colors: open, inactive, and closed states of the hERG channel. So with the Markov, you can introduce basically every single channel into the model and make it operational. At the moment, we have only hERG. So the hERG channel activity, according to what kind of action potential you measure, will be deconvoluted. When is it open? When is it closed? And when is it inactive? So basically, that will give you, or give us, a fingerprint once we have all the channels included. It sounds trivial, but when we got this far, the person left for Pfizer, who was doing it, and the other person left for something else. And we lost basically two people, so we are recruiting and building up this group. But the idea was here that, yeah, let's have mature cardiomyocytes, number one, then have a technology, which could be a high throughput technology, I come to that, and then have a model. So where are we going? Are we going for a fingerprint? Are we going for potencies? Or are we going basically for a kinetic description of what happens in these cells and how does it relate to early after depolarization? And I inserted this picture that basically it describes how theoretically the blockers could act at the ion channel. In this case, the blocker could go through and basically would not accumulate in the ion channels because this is the non-trappable blocker. But with the trappable blocker, it doesn't go further than basically stuck into the cells. And the difference between the K off would be important, that for non-trappable it's about 2 seconds, and for the trappable it's less than 0.1 second, which makes a big difference. So this is from the paper which I mentioned before, and there is now a major effort to find out whether this really in real life would materialize. This is, as I mentioned, this is theoretical at the moment. So what we thought, and this is now the other element here, to going towards a more complex system and develop something which would be used routinely for pro-arrhythmia assessment. So we were working briefly with QState, with the cardiac optopatch technology. So very briefly, this is a broad field microscopy where the cells in the plate are transfected either by a protein which allows spacing by laser, and then the other cells are basically infected with 2 other proteins, one sensing basically membrane potential changes or voltage changes, as listed here, and the other one is sensing calcium. And it's different fluorescence, so they don't interfere with each other. The interesting thing about it, that these cells, which are basically the pacemaker cells, the armored pacemaker cells, are sitting in one corner, and all the rest of the cells in the whole field. So apparently you don't see the pacemaker cells in real life when this is going on, which gives you an interesting element, which is the vectorial element, and that's exactly how the heart works. So that's, I think, is missing at the moment from many of the other experimental setups. So what you measure here in these cardiomyocytes, it's basically, this is the normal action potential, and then I know that if you and we were recording single cells, you see this is not an action potential. This is an average, in this case, of the action potentials. So that's why the distortion of the shape. And this is, this is the voltage in the presence of E4031, and this is the calcium components. But what I was really struck when these experiments were done, so there is a signal here, but it's an unusual signal. And when you look at the effect of, these are 5 different runs, and you employ basically only the MSO, there's a control. But what they did, they were pacing these cells. And at a certain point, the cells gave up on pacing, and they had some, not arrhythmia, but they were not following exactly the pacing, and then they recruited again and then went on. And for some reason, it was around 15 or 20 pacing, and then they did not follow. And that was pretty similar in all of the experiments. But you can see there are some changes in the action potential height. That's something which they considered not so significant. And indeed, they did hundreds of recordings. That was fine. But when you go to E4031, I think I timed this thing, and that's why I jumped. So here you see that with the increasing concentrations, shorter and shorter period of time, they lose their following of the pacing. And here you can also see a kind of early after depolarization event, which we observed in many of the other experiments as well. What really bothered us was the action potential size, because here you don't know what's the membrane resting potential, but the system will sense, obviously, the amplitude of the action potential. So that was diminishing, and one major reason for that could be that the resting memory potential shifts. So here we go, bang. Basically, UV block HERG, the resting membrane potential partially maintained by HERG, it's gone. So how could I assess this experiment at the end? It was a very useful exercise for us. We learned something. We hoped for a publication right away, but I think this is negative data in that sense. So we cannot say that this is a model. But it highlighted this problem with it. And we tried to go the right direction and make these cells much more mature. So expression of Kv 2.1 should be much better. And then hERG should be free from keeping the resting potential. We also tried, and I'm very honest with you here, to cluster these. So what we did, we basically have very similar parameters which others are using, except something with the conduction, so the spread of the activation. And then the clustering showed something. I won't take you through that. You can have, I think, access to the slides. But it was like domperidone was the largest effect, and we observed early after depolarizations, amiodarone and DMS, so minimal effect as you would see in the clinic. But this is so minimal number of data. I mean, a cluster of 2 honestly doesn't make tremendous sense in terms of statistics. And we didn't do more compounds. Originally, this was designed whether we could see trappable, non-trappable. But we did not go that far. And we certainly didn't see anything which would discriminate those compounds which were designated based on the oocyte experiments to trappable and non-trappable. So we didn't follow up on that. So the key findings were that the optopatch technology is really nice because we can do in parallel voltage and calcium transients. You can say MEA can do the same, certainly. Motion and conduction velocity. It can detect QT prolongation and abnormal waveforms, AEDs. And the compounds tested didn't show differentiation into trappable and non-trappable, as I mentioned. The advanced analysis algorithm combined multiple measures into assessment of proarrhythmia risk. That's wishful thinking in this case because we don't have the right data. But it seems to be feasible. It allows acute and chronic pharmacology. You can pull it out and put it back as many times as you wish into the microscope. It gives spatial resolution. Individual cells and heterogeneities can be measured. They can basically focus on a single cardiomyocyte and have that particular element measured. And the concern was the maturity of the cells. Changes in action potential morphology was an indicator of lack of maturity. It was easy to measure. And hERG blockers can cause action potential amplitude changes due to depolarized level. So that's what we are doing now, jumping a little bit at the moment, and most of the companies that we measure individual currents, the major lineage is the hERG, and we use the Q-patch for this measurement, and the primary assay is basically QSAR, and then we have radioligand binding, but that depends on the company's approaches. And then the major channels. What we know based on our experience that, for example, sodium channel activity, we see about between 1% to 5% in all together. Novartis makes about 35,000 compounds per year. That's 2011 data. And calcium we see rarely, and KCNQ very, very rarely. So we don't do that regularly. We have descending numbers for these tests. Sorry, I was going backwards. So basically the dream is still here, that if we can develop iPSC CMs in a toolbox, then probably we can replace what's in the background. But certainly there could be an argument about keeping individual channel assessments just for even for nothing else but validation of the data, and certainly at the very beginning of introducing something, this robust change. So the overall objectives is going back to where we started from, the high purity, either atrial, pacemaker, ventricular cell cultures, and how to combine them really. And that would have the structural functional equivalence to adult cells. The culture should resemble native population. Some people are playing with 3D cardiomyocyte cultures. We don't have experience with it. So that's something which we can look into. But then the measurements have to be done in a different way. And the in vitro disease models obviously are interesting to implement, because then we could study basically patient population characteristics in vitro before going to the clinic. Cell validation, we talked about it, and I mentioned. So the final element is develop a business model for production and implementation of the model. So that's also in our IMI proposal, and we discussed it with people from SIPA, with SIPA leadership to say so, and try to basically fit into this particular picture. So I just wanted to say some acknowledgement to people. Bob Perstein I mentioned, and a couple of people who are working in our group. And I don't have here Martin Trebert. His name was on the front page because we did many of these studies together with him. So thanks very much, and I'm happy or unhappy to answer questions.
So sort of in interest of time, why don't we take 2 questions?
It turns that we are similarly thinking, I think, all of us, in many ways, and nobody has a single answer. At least the people I talk to would not name names, but it was like a hope and frustration coming through the community during the discussions and during the past couple of months. I can tell you that when we went for the IMI, we said we need $15 million basically to work on this during the next 5 years. But then they asked for in-kind contribution. Thanks. In-kind contribution is not coming easy these days from industry because there are so many changes. And that's where we are at the moment, at how to make a next move and basically put this into operation. But I think SIPA is also converging all on these questions, certainly, and without it, it won't be a SIPA, an assay which is viable. I didn't talk, and that will be in the next talk, about the application of high throughput technology. With the QState, I touched on it, but there are various elements here, and we would be more than happy to see whether we can, for example, automate the individual cell recordings, which would be very important if this system really works as we hope it would, to decipher the kinetics of the channels. That would give a tremendous support, because then we would have a large bulk amount of data available at our hand, and then we could see variability also.
I had one question. Maybe you can comment on where we are in terms of reaching a more mature iPS-derived cardiomyocyte, and in your view, is any spontaneous cardiomyocyte acceptable in terms of the toolbox you're trying to develop?
You mean spontaneously active?
Right.
Ventricular cells? I would love to see quiet cells, which then can be activated. But I think that leaving cells quiet and sitting in the culture is not an option. So my ideal is that we have pacemaker cells, and they can live together with the cardiomyocytes from the left ventricle, with the left ventricle specificities, and they could talk to not talk to each other, but they could be paced, and they could take their identity, which is a difficult thing.
Mm-hmm.